Mordecai. I'm an associate professor in biology. It's such a delight to be introducing Marissa Childs today for her PhD defense. Marissa was one of a group of the first 3 students that ever joined my lab, and it's just such a huge honor that she was willing to take that risk on a new lab, and it's been such a joy to work with her over the years. Marissa came from a background that was not so traditional in my lab. We're a disease ecology group, and Marissa's background was in applied math, econ, environmental studies, honors in all 3 from her undergrad work. And she came into E-IPER saying that she really had no idea what she wanted to do, but was interested in trying to use these amazing quantitative and mathematical skills that she has to try to solve some of the really thorny and important environmental problems in the world, ranging from water to smoke to disease and many other environmental problems. And despite saying for many years that she had no idea what she wanted to do, that's basically exactly what she's done in her dissertation, and is now, despite her insistence to the contrary, I would say a full-on disease ecologist, among many other things. Marissa is this really rare person who has this really deep insight into thinking about biological and social processes in her work, but also having really strong quantitative skills and ability to really rigorously think through and use many different tools from math, econ, statistics, computer science, all over the place. She's really just an incredible student. And as a result, she's worked on a wide variety of different projects during her time in the lab. She's worked on, as you'll see, some of her projects are on yellow fever transmission in the Amazon and on effects of gold mining on malaria, but she's also worked on smoke pollution, on COVID transmission, on pathogen spillover of other wildlife pathogens, and many other things. And often the toughest challenge for Marissa is, given that she does have so many great skills and interests, like figuring out when to say no to all these cool opportunities that she's doing. So you'll see her research program is already a very well-developed, full-blown research program, well beyond what you see with most PhD students. Beyond all this, Marissa has been a really great friend and colleague and collaborator and mentor to so many people in the lab, ranging from people who are her peers, undergrad students, to undergraduates, to postdocs, and to myself. We all just really value Marissa as a lab member and as a collaborator. And as an advisor, it's been really amazing to watch Marissa, over the years, her confidence grow and her knowledge grow, and really to become a very confident and independent scientist. So it's pretty amazing to be saying goodbye to Marissa. She's going to be moving on to a really prestigious Hughes Environmental Fellowship at Harvard starting this summer, where she's going to be further pursuing some of her research interests in how environmental change affects infectious diseases as well as air pollution. And it's just so exciting to see where Marissa's going to go. So it's a delight to introduce Marissa Childs for her PhD defense.
Thank you for that introduction, Erin. I'm really excited to share with you all today my dissertation work on environmental change in human health. We all know that anthropogenic global change is rapidly altering the planet. From 2015 to 2020, an estimated 10 million hectares of forest were cleared each year. Simultaneously, climate change has increased global surface temperatures by about 1.1 degrees Celsius since the 20th century. We know that these changes have enormous health impacts, but it turns out we actually don't understand where and when they happen. The face of climate change is usually these Arctic sea creatures whose ecosystems are being destroyed and who are often facing extinction, but equally problematic is the human health toll of climate change. Unfortunately, we really don't understand these effects in many cases. And in order to enact policy that can help mitigate these effects, we need to understand where these changes are happening, who is affected, and how mechanistically the global environmental changes are affecting human health. So this leads me to ask, what is the scope of and mechanisms by which global environmental change is affecting human health? This is a difficult question, and there's a few reasons. First, we know that these effects are context dependent, meaning that local case studies that we do have don't always scale to global effects. For example, here's an example from vector-borne disease transmission. You can see increasing rainfall on the horizontal axis, and we expect that with more rainfall, we're going to see increasing vector breeding habitat, and so more likely, we'll be more likely to have outbreaks. But we also know that with extreme drought conditions, people store water. So we might also see more vector habitat and more likely to have outbreaks with droughts. But this is also dependent on the situation. In places where there's a lot of water available or reliable access to water, people may not be storing water, and so we might not actually see that breeding habitat. In addition to these context-dependent effects, we also know that there's multiple simultaneous changes. There's land use change, there's climate change, there's increasing human mobility as well as increasing human population. And on top of this, we're almost always limited in the availability of on-the-ground data, especially in the places that are most affected by these changes. So my approach is to use data-driven models that incorporate both ground and satellite information, to examine over large spatial scales these effects while accounting for the local conditions in those places. My research and this approach would not be possible with a lot of existing infrastructure. In particular, I leverage foundational hypotheses about how environmental change affects human health from early case studies. We also take advantage of data collected by governments over large spatial scales or long time series. And these kind of data wouldn't really be possible for a single researcher or even a single research team to collect. We can also integrate these data with satellite imagery. The recent explosion of high-resolution satellite imagery and the computational power to store and process these data has made it possible for us to quantify the effects of global change on a really high resolution and local areas. And finally, complementary statistical advances have improved our ability to integrate these different components and to attribute the effect of global change on human health. My research looks at 3 different projects. The first, on vector-borne disease, looks at the effect of gold mining on malaria. The second turns to look at yellow fever spillover. And the third focuses on pollution from wildfire smoke. So for this first chapter, gold mining's effects on malaria. Malaria is a really deadly disease that's pretty widespread. There was, WHO estimated that in 2020 there were about 200 million cases. But in South America, where I'll be focusing, we're nearing elimination in some regions. Malaria is transmitted between humans and mosquitoes. And for a long time, we just kind of thought of these mosquitoes as flying syringes, just transferring the virus from one person to another. But we now know that that's not the full picture. These mosquitoes actually have really intricate biological processes that are happening inside of them. And simultaneously, these mosquitoes are going through their own life cycle. They lay eggs in aquatic breeding habitat. Those eggs develop into pupae and larvae and emerge to new adult mosquitoes that can go on to carry malaria. So given this life cycle of mosquitoes, it's understandable why environmental change might affect malaria. You can see in this image gold mining in Brazil. And some of the really important things I want to highlight in this image are first the small ponds created by mining that are near forest edge. These create really ideal habitat for mosquitoes, particularly the Anopheles darlingi mosquito that transmits malaria in Brazil. Second, you can see in this lower corner, human settlement. These people who are working in the gold mining areas often are living in really close proximity to where these vectors are with limited access to healthcare. And finally, what you can't see in this image is that the miners are often a pretty mobile population. So when we see increasing malaria transmission in a gold mining region, we might also expect to see that spill over spatially into new geographic areas. So we have these 3 clear hypothesized mechanisms of environmental change, occupational exposure, and human mobility. And while local case studies support these mechanisms, we actually have really poorly quantified total effects of gold mining on malaria. So this led me to ask, how much does small-scale gold mining affect malaria transmission? And do these effects extend beyond the location of gold production? As you can see by this kind of remote nature of this gold mining, it's really difficult to measure, especially because small-scale gold mining is often done informally. One place where we do have data to study small-scale gold mining is in Brazil, where the government collects data on gold production via the taxes that they collect on gold transactions. Here you can see the pattern of where gold taxes were collected. And in particular, I'm showing you the Amazon region of Brazil where malaria is primarily transmitted. In Brazil, the two main parasites of malaria are Plasmodium falciparum and Plasmodium vivax, shown in red and blue, here with higher incidences indicated by darker colors. You can see that the spatial pattern of Plasmodium falciparum and Plasmodium vivax generally match the spatial pattern of gold production. But we also know that this spatial pattern is shared by a suite of other covariates that are also expected to affect malaria. So to disentangle these effects, my approach uses a Poisson regression where we estimate the effect of gold mining on malaria by including municipality or unit fixed effects. In doing so, we identify the effect of gold mining on malaria by comparing each location to itself over time as mining activity fluctuates. We include state year fixed effects that can account for common regional changes, and we also include other local covariates to control for their effect on malaria. We include both local and distant gold production in this analysis, given our hypothesis that there might be local effects as well as spatial spillover. I mentioned that we have these two parasites, and one of the interesting biological differences between the two of them is that Plasmodium vivax has a liver stage that can cause relapses months or years after the initial infection. This means that infected people are more likely to be able to travel and spread the disease. So we expect that both parasites will show local effects, but primarily Plasmodium vivax will show distant spatial spillover effects. We run this analysis for municipalities in the Brazilian Amazon from 2010 to 2015. So what did we find? First, we're going to look at the local effects. Here you can see gold production on the horizontal axis and malaria incidence on the vertical axis. The solid lines indicate the estimated effect of gold mining on malaria, with the shaded area showing the 95% confidence intervals. Plasmodium falciparum again is in red, and Plasmodium vivax is in blue. For both species, we see that increasing gold production is associated with increasing malaria incidence. We also can see that we estimate these effects plateau, with the greatest effect at early stages of mining where really small quantities of gold are being produced, and much smaller effects at higher quantities of gold production. Next, turning to the distant effects. Here we can see distance from production on the horizontal axis rather than the quantity of production, and on the vertical axis I'll show you the percent change in malaria incidence. For Plasmodium falciparum, as we hypothesized, we see those clear local effects, but no effects in further out distances, For Plasmodium vivax, on the other hand, we see effects both locally and up to 250 kilometers away. So given these local and distant effects, we wanted to understand what the impact of them was. While we see that the marginal effect of a local change is a marginal effect if a change is slightly larger locally, an effect that influences municipalities up to 250 kilometers away could potentially be affecting many municipalities. So we can estimate the effect locally and distantly by simulating what would happen if we increase gold in places that are already producing it. Here I'm going to show you the additional cases if we simulate an additional kilogram of gold production. First, looking locally, each point here shows a municipality where gold was produced and the estimated increase in malaria cases in those locations. On average, we're estimating about a half an additional case for Plasmodium falciparum and about 2 additional cases for Plasmodium vivax. When we look distantly for, sorry guys, you're getting things cut off here. I think this slides aren't quite fitting on the screen.
Not too bad, we can see.
Okay, so distantly for Plasmodium falciparum, we can see that about 0 effect given the non-significant effects for Plasmodium falciparum. But for Plasmodium vivax, we're actually estimating an additional 58 cases in distant municipalities. So these distant effects can really exceed the local ones. So what have we learned from this? First, there are clear effects of gold mining on malaria in the municipality of production for both species. And consistent with the differing parasite biologies, there are effects up to 250 kilometers away, but only for Plasmodium vivax. We also find that spatial spillover is a really important consideration for malaria control. Given these spatial spillover effects, places that are implementing malaria control efforts could really see their efforts undermined by the spatial spillover from mining areas. Finally, I just want to point out some next steps we have going on here, which are to extend this data set up to 2019 and to additionally use a remotely sensed measure of gold mining in addition to our tax-based one. Moving on to the second chapter, we're going to talk about yellow fever spillover, also in Brazil. Unlike malaria, yellow fever is primarily transmitted between non-human primates and canopy-dwelling mosquitoes in the sylvatic or forest cycle of transmission. It's only through spillover from this transmission cycle that we see human cases of the disease. Yellow fever is this really interesting disease because we've studied it for such a long time. You can see from this 1856 map of diseases that there were many areas where we knew yellow fever was transmitted. It was the first disease ever identified to be transmitted by a mosquito, and it's been studied for over 120 years. In 1937, we developed a really highly effective vaccine for yellow fever, but despite this vaccine, we still see outbreaks. And in December of 2016, we saw an unexpectedly large outbreak occurring in the southeast coast of Brazil. This outbreak happened both in known areas at risk of yellow fever transmission, but also in new areas highlighted in the dark green. Kind of scarily, yellow fever has a really high case fatality rate, 20 to 60%. So understanding what drives this spillover is really important. So what do we know about spillover? We know that spillover can only happen when conditions align in the primate reservoir, the mosquito vector, and the human population. And we can think of these aligning conditions as layers in space and time. It starts with an infected primate. There must be mosquitoes in sufficient abundance and proximity to take a bite on an infected reservoir host. That mosquito must then survive long enough to develop the virus and travel far enough to come into contact with a human. That human further has to be susceptible to the virus and then report as a case in order to be identified as spillover. I want to note that this spillover process, even when all of these conditions align, is inherently stochastic. There's something random about when and where exactly a spillover case will occur, which makes prediction really difficult. But we also know that prediction is important because if we have early warning of when spillover is going to happen, we can use the various public health tools, including mass vaccination and vector control, to prevent those cases. So given this conceptual model of yellow fever spillover, how can we integrate our understanding of all of these component parts that we've studied for so long to estimate and predict spillover? We incorporate our mechanistic understanding from all of these layers and we'll construct a mechanistic model of pathogen spillover. But we know that whatever mechanistic understanding we have, however long we've worked on it, is going to be incomplete. There's parts that we're still missing. So our second question is, what additional drivers are we missing in this model? For our first question, our approach is to synthesize our understanding of the individual biological processes at play here, and then to integrate these to create risk metrics. We'll estimate those risk metrics for 2001 to 2018 and compare to human cases of spillover. So for that first step, synthesizing our understanding of biological processes. I'm not going to do this for all the layers, I just want to highlight a couple that are somewhat interesting ones. For vector distribution, we take advantage of all of the vector captures that people have been spending so much time doing over the years, and we use hundreds of mosquito captures to estimate the environmental covariates that predict where these vectors occur. We also use information from another really cool old study from the 1940s to estimate how far mosquitoes can travel. This is a study from the 1940s where people captured mosquitoes, dusted them with bronzing powder, re-released them, and then waited to capture them again. And they spent thousands of hours waiting to capture these mosquitoes. With this, we can come up with a pretty clear idea of how far these mosquitoes can travel and how likely they are to re-encounter another human. So we do similarly for all of these other pieces, and we can come up with estimates for each of these of what biological processes underpin them and how we think they depend on other conditions. We can think of each of these layers as like a probability. And so to calculate the probability of spillover, we can then multiply all of those to get a joint probability, and we'll integrate over all these layers. We can calculate 3 different risk metrics. For the first one, we include the layers for which we know environmental conditions affect spillover probability. We call this environmental risk. We then continue on adding on top of environmental risk, human susceptibility, and incorporating information on human vaccine coverage. We finally incorporate information on human distribution and come up with what we call population scaled risk. So now we have these 3 risk metrics, and we wanted to know originally how well can our risk metrics, based on this mechanistic model, predict spillover. To do this, we compared human case data. Here I'm going to show you the different risk metrics on the horizontal axis and predictive accuracy on the vertical. I want to recall that spillover is this really random process, so we don't expect to be perfect at predicting. Here on the vertical axis, our predictive accuracy is measured where 50% is as good as random and 100% is perfect distinguishing. Perfect, yeah, let's call it distinguishing. We can calculate the average risk metric for each municipality month and compare it to whether or not that municipality month had a case of yellow fever. Surprisingly, we see that environmental risk does best, which is unexpected given we're incorporating additional information into our immunological and population scaled risk metrics. This suggests that these additional covariates aren't helping us to predict where pathogen spillover will occur. While we've first here calculated mean risk, we know that the processes between and the interactions between primates, vectors, and humans are happening at a scale that's much finer than a county or municipality. So we can also calculate maximum risk in each municipality, which might better capture the very local alignment of conditions. Interestingly, we find that maximum risk does better across all of our risk metrics, suggesting that it's really local alignment of conditions that is important for spillover. We find that even our best risk metric, maximum environmental risk, has about 72% prediction accuracy. So we're still missing a lot here. This led us to ask, what additional drivers can we use to improve our predictions of spillover? Our approach here is to use current and 1-month lagged environmental risk and to add in additional covariates that we think are associated with spillover. We use these in a machine learning model called a boosted regression tree to predict occurrence of spillover. Boosted regression trees iteratively fit regression trees, each time fitting to the errors of the previous tree. Boosted regression trees are really well suited to this kind of problem because they allow us to estimate both nonlinear effects between a covariate and spillover probability, as well as interactions between different covariates. In our boosted regression tree model, we find that our out-of-sample predictive accuracy is about 95%. We can now dig into this boosted regression tree model to look at which variables are most important. Here I'm showing you relative importance on the horizontal axis as measured by gain in model performance from that covariate, and on the vertical axis we have the different risk metrics. Here I'll highlight our 1-month lagged and current environmental risk, shown in red now, which we can see are the second and fourth most important predictors. Zooming in on these, we can look at the estimated relationship between each covariate and spillover probability. Here the environmental covariates on the horizontal axis and spillover probability, or the marginal effect of spillover probabilities on the vertical axis. We can see that at higher risk metrics, we have a higher probability of spillover. And similarly, looking at current environmental risk, we find a similar relationship. So our environmental risk metric is capturing the relationship we think it is. But there's 2 other important ones here. I've just shown you the second and the fourth, but the first and the third are actually more important than our environmental risk metric. And these 2 are temperature and vaccine coverage, now shown in blue. For temperature, when we look at the marginal effect, we find a higher probability of spillover at low temperatures, suggesting there's something happening at those lower temperatures that we haven't captured in our mechanistic model. For vaccine coverage, we find a kind of unexpected relationship, where higher vaccine coverages are associated with higher probability of spillover. There's a couple possible explanations here, one of which is that public health officials know when spillover is likely, and so they're vaccinating people. So we might actually be detecting a reverse causal relationship here. Alternatively, places with high vaccine coverage may have really good public health infrastructure, and so we may be more likely to detect spillover cases when they do happen. In either case, this explains why our immunological risk actually performed worse, because incorporating our naive expectation of how vaccine coverage expects spillover worsened our predictions. So to recap, we found that environmental conditions constrain spillover. We also found that it's really local variation in these environmental conditions that's important for when and where we can see yellow fever cases. In our statistical model, we found that temperature and vaccine coverage were important predictors that we may not have well captured in our mechanistic model. And we also find that 1 month lagged environmental risk is an important predictor for spillover, suggesting that there's the potential for that kind of forward prediction I was talking about earlier, where we can use prediction to inform public health interventions. Now I want to pivot away from vector-borne disease and think about wildfire smoke instead. For anyone who's lived in the Bay Area for the last couple years, you might remember this day from September of 2020 when the sky turned orange. This happened from a combination of wildfire smoke and fog over the Bay Area. And anecdotally, anyone who lives in the Bay Area will probably tell you that wildfire smoke has been increasing. This is due to decades of fire suppression as well as a warming climate. But these problems aren't limited to the Bay Area or even the West Coast. Here in the satellite imagery from NASA, you can see shown in red areas of active burning, and lofted high in the atmosphere away from those burning areas is wildfire smoke that's then carried on the jet stream all the way across the country. Here's New York City in July of 2021, where the ground air quality is being affected by wildfire smoke all the way from the West Coast. And we know that this kind of air pollution has huge health effects. One of the most important pollutants is fine particulate matter, or PM 2.5. 2.5 here refers to the diameter of the particles, which is 2.5 micrometers, which is about 1/30th of the diameter of a human hair for reference. We know that coarser particles, PM10, can get into the human body, get into the nose and throat and cause negative health effects, but we know that PM 2.5 can travel even further, getting all the way down into the lower respiratory tract and the lungs. It's estimated that about 60,000 premature deaths in the US in 2019 were due to air pollution. So when I started this project, I wanted to know, what are the health effects of this smoke PM 2.5? How do they differ from all-cause PM 2.5 health effects, given the different nature of pollutants and the often extreme effects? But as it turned out when I looked into this, we don't have good exposures of when and where people are being exposed to wildfire smoke. So we scratched that, and instead we asked, how much PM 2.5 from smoke are people being exposed to? This is a hard question. I want to highlight two particular challenges. The first is how we identify which portion of PM 2.5 is due to smoke. Here's the kind of data we often see, which is total PM at monitoring stations. But as you can see, this total PM doesn't help us identify which portion is from smoke. And we actually only have these monitoring stations at about 2,500 locations throughout the US. These are maintained by the Environmental Protection Agency. And as you can see, there's large areas in the US where we don't have monitors. So our approach to handling this proceeds in 3 steps. First, we identify days where the air quality is affected by smoke. Then we use those smoke days to infer what portion of PM 2.5 is due to smoke. And finally, we predict that smoke PM 2.5 over the contiguous US. For the first step, identifying smoke days. Analysts at the NOAA Hazard Mapping System hand draw plumes from wildfire smoke every day. But we also know that these plumes aren't a complete picture. You can see from the satellite image of the same day that clouds are obscuring much of the US and probably hiding where other plumes may be happening. So we can incorporate additional information using known locations of fires to simulate air trajectories from those fires. We can then find locations with densely packed air trajectories to identify what other locations might be affected by smoke. We can then use that classification of smoke days to infer what portion of PM 2.5 is due to smoke. Here's that time series I was showing you earlier of total PM 2.5, and we can bring information in on smoke days. We can then use those smoke days to calculate location and season-specific non-smoke medians. Here, these medians are essentially serving as a counterfactual. How much pollution would we have expected if there was no smoke in the air? And then we can attribute any pollution above that median to smoke, allowing us to define smoke PM 2.5. Now, taking this smoke PM 2.5, we can predict over the entire US. We use a set of spatially and temporally consistent inputs, and train a boosted regression tree model similar to the one that I talked about in the second chapter, and use these to predict smoke pollution on a 10-kilometer resolution over the entire US daily from 2006 to 2020. We can then measure how well this model is doing by leaving stations out of sample and comparing our predictions to what we observe at those stations. We find that our model explains about 67% of the variation at new locations that it hasn't seen before. I want to show you an animation of smoke from 2020, but to orient you, first I want to show you a little bit about how smoke, or PM 2.5 is measured. PM 2.5 is measured in terms of concentration, micrograms per cubic meter. For anyone who's thought about AQI before, the AQI that's in the unhealthy range is equivalent to PM 2.5 from about 55 micrograms per cubic meter and up. On the other hand, the EPA daily standard for what is maximum allowable is 35 micrograms per cubic meter. But this is a regulatory limit, and the WHO actually recommends an even lower limit of 15 micrograms per cubic meter. So you can see in this animation now that wildfire smoke is pretty variable over time, and in winter there's not much of it. Getting into spring, we see more fires, especially in the Midwest and the South. And starting in spring and summer, we'll start seeing fires in the West Coast. These fires send out huge plumes that cover large swaths of the US, and into August, we see these engulfing the entire country. At many times, the entire western half of the country is covered in unhealthy levels of smoke pollution. By November and then December, most of these fires have faded and the pollution with them. So I showed you that kind of terrifying image of how much pollution we're all being exposed to. But those are daily estimates, and they're pretty extreme, but they're also happening over a short time period. So we wanted to understand how much is this going to contribute to the average pollution that people are exposed to. Here we've calculated the average annual contribution of PM 2.5 to pollution levels. Here this is for 2006 and for all of the other years. You can see that the numbers are a little cut off, but that bright yellow is about 5 micrograms per cubic meter. These predictions really highlight the variation between years, with some especially bad years in 2017, 2018, and 2020, especially in the western states. To put these numbers in context, I said that the bright yellow, whose numbers you can't see, is at about 5. And while the EPA regulates that daily maximums are at 35, its regulation for annual maximums is 12. So this pollution from smoke alone is making up almost half of what the EPA says we should be exposed to in a year. Given these changes, we then wanted to quantify how much has smoke pollution changed over this time period. To do so, we calculate an average during the first 5 years and an average during the last 5 years and the difference between those. This gives us an estimate of the decadal change without overweighting on some of the more extreme years like 2020. We find that the decadal change in smoke pollution, as expected, is really concentrated in the western part of the country, with some places experiencing an additional 5 micrograms per cubic meter every year. To put this number in reference, I know I said this is about half of the EPA annual maximum, but we can also put this in terms of the pollution improvements that we've seen in the past. In the 1970s, the Environmental Protection Agency started regulating for the Clean Air Act, and we saw huge air quality improvements. Those air quality improvements are estimated to be about 5 micrograms per cubic meter over the past decades. So wildfire smoke pollution has undone those improvements from the Clean Air Act, essentially. These increases in annual averages are driven by both more smoke days and more extreme days. We can look at the days with smoke PM 2.5 over 50 micrograms, so in the unhealthy range, and we see that many places are experiencing an additional 10 days of year with extreme smoke, And as expected, these places are really concentrated in California, Oregon, Washington. We can then estimate the population exposed to these levels of concentrations. Here we've quantified the average number of people exposed to at least one extreme day. From 2006 to 2020, it was about 4 million people exposed each year. And I should mention, this vertical axis is on a log scale, so we can see these, because these numbers are kind of so crazy you can't see them on a regular scale. We estimate about 4 million people exposed during the early time period, and a 4-fold increase up to 16 million people in the 2016 to 2020 time period. 50 micrograms is bad, but 200 micrograms is even worse, and we can also look at that change. From 2006 to 2010, almost no one was exposed to this really extreme level, but from 2016 to 2020, about 1.5 million people were exposed every year to at least one extreme day. And this is a kind of crazy 11,000-fold increase. These views of smoke pollution have become far too common on both coasts. Our predictions show that these increases in smoke PM2.5 are undoing air quality gains from decades of regulations. And kind of scarily, we also know that wildfire smoke is exempt from air quality regulation, as it's defined as an extreme event. So there's no policies currently in place to regulate this extreme exposure. Our next steps in this project, or in future projects, are to link these exposures to health outcomes so we can identify who's most exposed and how their health is being affected, which will allow us to better target policy. I've shown you these 3 projects that I worked on throughout my dissertation. To recap, in the first one, looking at gold mining's effects on malaria, we saw that gold mining increases malaria locally and up to 250 kilometers away. In the second chapter, we looked at yellow fever spillover and found that local variation in environmental conditions predicted spillover risk. And in the third, we saw that wildfire smoke is undoing air quality gains from decades of regulation. So looking at these 3 chapters together, a few themes come through. First, environmental changes have a huge health toll. There are pervasive health effects that spill over far beyond the location where a fire is burning or where gold is mined. But to enact policy, we really need to understand where, who, and how these changes are happening. And the kind of analyses that will help answer those questions are these kinds of large-scale spatial analyses that simultaneously consider really important local variation. These kind of analyses and this work would not have been possible without a bunch of people, and I want to take a moment to acknowledge them. I'm going to be brief so I don't get emotional about any of them, but the E-IPER staff do an amazing job at what they do, and their dedication and hard work really is apparent to the students, and thank you guys. I have 2 awesome set of lab mates. They have both taught me so much about science and how to do thoughtful science, but also been so much fun in the process. They've also answered my questions that I send out at weird hours in the morning about what kind of ticks my dog has collected. I've also been really fortunate to work with an amazing set of collaborators and mentees, and I've really learned so much from them in the process. I started this program with 7 other people in my cohort, and I think they're one of the reasons that I started this program, and they remain one of the reasons I'm excited about the work I do. My advisors in committee, Steve, David, and Desiree, the research that you guys do is so inspirational, and you're also such thoughtful humans. You really make me want to keep doing research. Marshall and Erin, thank you. You guys have taught me so much. I don't have words for it, so thank you. To friends and family, you've put up with all my PhD angst and also reminded me that it's important to go out and do fun things. So thank you all very much. I really appreciate it. Thank you. I did get emotional, sorry guys. I'm happy to take questions.
Background question. When you were giving the death toll for the pollution exposure, I'm assuming that's kind of like across a variety of causes. Can you talk a little bit about like what that is, like asthma or lung cancer?
Yeah, so there's a bunch of things that we know smoke or pollution affects in terms of health. Exactly as you said, there's the kind of respiratory effects like asthma. There's also cardiovascular effects. I think there's also a lot of effects we don't understand. And so sometimes these estimates include like inferred effects over the entire population where we estimate all of the ways that people have died and then simulate basically if we reduced pollution levels to some plausible minimum, how much would deaths be reduced? And that allows us to quantify what portion or how many deaths were due to air pollution. So I think you're exactly right. There's those set of deaths, or those set of causes, and then a whole bunch that we're still trying to figure out today.
Great.
Right, so Kari asked online, first she has a lot of compliments for you, but I'll skip those. You want to hear those as well. Fantastic presentation. I congratulate you on your work and your thesis and your wonderful mentorship through your time at Stanford. Good luck with Boston. I had one question. Given the results you showed on the exposure to extreme days of wildfire smoke, for example, from 2016 to 2020, what about the dimension of cumulative exposure over time, years of exposure?
Yeah, I think that's a great question, and one we don't really understand. Kari's work actually starts trying to unpack that, and I think they've done a great job of that. That's what we're hoping to do next with these estimates, is use them in health studies to try and understand both the short-term effects and the cumulative effects of these kind of exposures.
So maybe another way of interpreting the question is just how much has cumulative exposure changed for people?
Ah.
I imagine it's even more than the annual exposures because
Yeah, so it definitely will build up over time. We haven't quantified that cumulatively. I guess you could think about that in terms of if you visually add up all those maps over the years. For anyone who lived in the Bay Area through all that time, you've been exposed to all of that, unfortunately. So we can certainly start doing that, and I think that it will be part of the process of quantifying these health effects. Thanks for the question, Kari.
This is not like exactly related to the research you presented, but like you said, we've been exposed to these effects if you walk outside, but how much can indoor air quality mitigate the effects of wildfire smoke on pollution exposure?
Yeah.
If you stay inside and have the filters on?
That's a great question. I think we know that, especially in well-sealed houses, that you can be buffered from these effects by housing. But we also know that there's a bunch of people who either don't have homes or don't have homes that are well sealed. Those are the set of people who are also exposed to kind of extreme temperatures as well. And those people will be now like a compounding of these problems. So it's an unfortunate case where it's probably only going to exacerbate the inequality we see. I think you are likely better in your house than outside, but we also know that pollution can infiltrate into houses. And so that's where the filters will really help to reduce indoor air pollution. Yeah, Marshall.
Chapters 1 and 2 were on infectious disease, as best I understand it. Chapter 3 was not yet on any sort of health outcome, but it's pointing probably more towards non-infectious stuff. How do you see the interaction between those chapters? Do you think the third exposure could be useful for questions of non-infectious disease?
Yeah, definitely. I think people have even done that already in the ongoing pandemic. People have estimated that exposure to pollution and wildfires has increased COVID and COVID mortality. I think this is still an open question. Unfortunately, we often segregate environmental health things into like a global health infectious disease category and an environmental exposures or environmental epidemiology, and we don't really like to mix them very much. But these kind of global changes are going to affect all the components, and so it's kind of important that we start thinking about them together so we can better understand how these kind of environmental exposures will affect infectious disease as well.
That's a good thing for a postdoc.
Worried who's going to ask which advisor works on more important health.
That's a good question.
Which of these, if you add it up, seems more important?
All right, I think we have like 3 minutes left if there are questions. I don't see any online. I know a lot of students are wondering how she made those great visuals, but they can ask her that later.
I can ask you in general, and then it'd be fun. So you're bringing up environmental justice, and so a lot of what you've talked about is environmental change, which is linked in with environmental justice issues, global health inequities, and all of the systems that you're talking about are completely impacted by that. And now you're talking about how effects on those who are most vulnerable are going to be compounded, right, exponentially as this continues. So just dream a little bit with me about how your work and your life's work studying these systems is gonna go about to drive some of the changes that need to happen.
Whew, okay. Yeah, I think that's a great question, and I think definitely the research I hope to continue doing will look at this. I think these chapters haven't gone far enough to address the justice question. I think they've pointed out how environmental change and health are related, but they haven't really tried to link that to the existing vulnerabilities that people have, the existing health conditions, and the societal forces that have left them in that state. I think it's important to start considering all of those together. And I think we need to work with both those kind of communities who are affected and policymakers so that we can create the policies that ensure that these devastating effects don't fall on their shoulders. I'm not sure I dreamed big in what I would do there, but I think that's definitely what I'm hoping to continue working on in my postdoc.
Okay, well, I guess we can close it out there and